Illumina sequencing machine, but I'm using a method that now is kind of the trend, or try to be the trend for population genetics studies, when you want to use many regions of the genome for each individual. In 2010, 2011, they published this method that what it does is it targets regions of the genome that are surrounding restriction sites. I don't know if I'm getting too technical. No? Okay. So what happened before is that if you were sequencing whatever DNA you had in your extraction, there was little chance that you will get the same regions for two samples if you were doing it randomly, because the genomes are so wide that it's difficult to get the same region. That is what you need if you want to compare sample A with sample B. No? So the option is do full genomes, of course, but when you don't have so much money and you want to do a lot of samples from different regions, then it's not feasible because it's too expensive. So they came with this idea that it was you get your DNA, then you digest it with an enzyme that what it does is it cuts the DNA in a specific place. So if you have sample A and sample B, you digest both samples and both will be cut hopefully in the same places. And then, as you know that you have a cut inside there, you can add a series of adapters and this protocol to try to sequence the sites that are around those restriction sites. So then you increase a lot the chance of getting the same region in a number of samples. That is what you really want. So I want, for example, to compare 50 different dolphins, so I want to get the same areas. An old technique. Now, what they use from the old technique is the digestion with enzymes, but then they use next generation sequencing to get the sequences, right? Rat sequencing that hasn't been used before in marine mammals. This is also an advance and something exciting to use it for the first time in a new animal. But anyway, I think the key of my project, maybe it's because I'm a marine biologist or ecologist, I'm more interested in the application of this, what will you get, than in the technique. But of course, I think you had a lot of people from geogenetics, and you see that the technology nowadays is so important and it's evolving so fast that it's great to be able to use the latest advances, of course. I want to understand, of course, what I do with every step. And I also, once you get your sequences, it can also be a black box, because you get millions. And then if you're just doing the lab work and someone else is analyzing your data, it's the same. It can be a black box afterwards. Like they tell you, oh yes, we have found that the Icelandic dolphins and the Danish dolphins are totally different. And you just say, okay. You really need to understand the whole process. But it's true that I'm more interested in the biology or ecology of the organism than to know if the DNA breaks because the bonds between molecules are less strong or more strong. So it's good to know, but it's not what catch my attention.